Hi everyone, and welcome to our screencast all about DNA and DNA replication. So before we get started, let's talk about a few things to do with DNA. First of all, it's really interesting to think that 50% of our DNA is exactly the same as the DNA in a banana. And if you took your DNA and stretched it out end to end, it would go to the sun and back 600 times. There's about 9 feet of DNA in every cell in your body. And 99% of your DNA is identical to mine and identical to everyone else in the class. So there's only about 3 million nucleotides that are different between you and everyone else. And your DNA can store more information than computers can. And when you eat a meal, you eat almost 93,000 miles of DNA at every meal. In the next 60 seconds, in the next minute, your body will make enough DNA that if you linked it together, it would stretch 100,000 kilometers. So your body's constantly making and replicating DNA. If you look at these chromosomes, this is a great picture of all of the chromosomes in one cell in your body. There's 23 pairs and 46 altogether. So by the end of this unit, you need to know what the 4 bases of DNA are. And we already know they're adenine, cytosine, thymine, and guanine. And then you need to know what a nucleotide looks like. We've already talked about that. You also need to understand how DNA is replicated with their 3 steps, and you need to know what recombinant DNA is, and the at least three uses for recombinant DNA. We'll talk about a lot of them. And then of course you have to contrast DNA and RNA, which you've already done. So you have a whole bunch of vocabulary words to know and understand. So let's go back to the beginning. What do we know? Well, we know DNA is made of nucleotides, made of a phosphate group, a sugar group, and a nitrogenous base, which will either be A, T, C, or G. So here's our pentose sugar. In DNA it's deoxyribose, and our nitrogen bases can either be purines or pyrimidines. And you'll remember, cut the pyramid. The ones that only have one ring have been cut, so that's cytosine and thymine, and uracil if you're doing RNA. And the ones with 2 rings, it's got 2 B gap, are guanine and adenine. So let's take a look at what a nucleotide is made of. You see here in this case, if this is adenine, it's attached to a pentose sugar and to a phosphate group, and that makes a nucleotide. We also talked in the last screencast about the fact that adenine always bonds with thymine, and they do that with 2 hydrogen bonds, and cytosine binds with guanine with 3 hydrogen bonds. And they all twist up into a double helix. As you can see here is adenine binding with thymine. You can see the 2 hydrogen bonds. And I want you to pay attention today to the fact that it's the sugar that's attached to the base. And the phosphate is in between 2 sugar groups. So if you look at the sugar-phosphate backbone, you'll see a sugar and a phosphate and a sugar and a phosphate, sugar, phosphate, etc. And that forms the sugar-phosphate backbone that twists up to form that double helix shape, as you can see here. Let's take a look at how DNA actually wraps in on itself so it can fit inside the nucleus. The yellow shapes here are histone proteins. You can see the double helix is being wrapped around tightly around those proteins. And then they'll get bound and twisted up farther in upon itself. And you can see chromosomes are huge, really, really long. And so if you want to get them into that nucleus, you'll have to condense that even farther so it gets all twisted up. And then coiled again into a chromosome. What is the job of DNA? Well, DNA tells the cell which proteins to make, when, to make them and how. So DNA gets copied into RNA, and RNA is translated by a ribosome into a protein chain. The second job of DNA is it stores all of the information that you need to make every cell in your body. So we all start with a stem cell, really, which can replicate itself, and stem cells can become any number of cell types in your body. So skin cells, nerve cells, blood cells, every type of cell in your body started out as a stem cell, which is why stem cell research is so big right now. And of course, if we want evolution to work, DNA also has to have the ability to mutate and change. And there's a lot of research going on right now with viruses and jumping genes, and they think that jumping genes in our DNA, which used to be considered as junk DNA, are actually viruses that have inserted themselves into our genetic code. So here you can see the correct copy of DNA, and it can be mutated in many different ways, and we'll talk about that in the next screencast. And of course, the last function or job of DNA is it can make copies of itself, and that's what we're focusing on today. So DNA replication is semi-conservative, and what that means is that the original strands, seen in blue here, remain intact, and what they do is they act as templates for the new strands that are being duplicated. So you can see in blue, those are the original strands, and each new copy of DNA has at least one of the original strands. And one copy of DNA will actually split apart, and half of the old molecule will be in each of the new molecules. So if you look at the two new molecules here, the blue strands are the new and the red are the old. So each new DNA molecule has at least half of the original parent. So what are the steps to DNA replication? How does this all work? The first thing that you have to do is you have to unzip the DNA. You have to cut those hydrogen bonds, and there's a special enzyme that does that, and that enzyme is called helicase. Now you can think of helicase as a big pair of scissors. Helicase will cut those hydrogen bonds between G and C and between A and T, and it'll separate one side of the DNA molecule from the other. See my little cartoon here, and if you were wondering how Bob got unzipped, it's because he was unzipped by helicase, cutting those hydrogen bonds. So it cuts the 3 bonds between G and C and the 2 hydrogen bonds between A and T. The second step that happens in DNA replication is complementary base pairing. So once the 2 sides of DNA are separated from each other, the new base pairs from the food that you ate come and join their partners. So anywhere where there's a T, an A will join. Whenever there's an A T will join. Whenever there's a C, a G will join. And whenever there's a G, C will join. And that takes a lot of energy, so you use ATP to build the new molecule. So if you had A in the original chain, then a thymine will come and bond to make a pair, complementary base pair. And if you had cytosine in the original strand, guanine will come and bond to make a complementary pair with cytosine. Next step is you have to join that all up with a new sugar phosphate backbone, because the new strand doesn't have a backbone yet. It's not held together, and there's an enzyme that helps with that, and that enzyme is DNA polymerase. And while it's joining the backbone, it's also checking for mistakes as it goes along the chain. So if you watch this animation here, you'll see the cytosine is joining the guanine, it's a complementary base pair, and the thymine is joining the adenine, and you'll see right here there's a bond made between the sugar and the phosphate, and DNA polymerase helps with that. So you get complementary base pairing, and then the backbone is formed. So it all happens kind of as you go, and there you have your whole new chain. Last step to DNA replication, of course, is that you end up with 2 completely identical strands of DNA molecules. That is, of course, assuming that there haven't been any mutations or any mistakes made. Let's take a look at what we mean by semi-conservative. Here is the original strand, and I'm coloring it yellow. So that's the original DNA, and you can see at the bottom it's starting to become unzipped. When you look at the identical strands, you'll see each of them has 1 yellow strand one of the original parent copies of the DNA strand. And you'll see I am drawing here in red the brand new strand that was just copied from the complementary bases coming in and joining up. So here you've got two identical DNA strands. Each DNA strand has one new side and one old side. Now all this DNA replication happens in what we call a bubble. So inside this replication bubble it's just copied a little bit at a time until the whole strand is copied. And of course there's two enzymes involved, helicase and DNA polymerase. Helicase cuts the bonds and DNA polymerase helps with complementary base pairing and joining that new sugar phosphate backbone. Again, the steps of DNA replication are you unwind with helicase, the complementary bases join up, a new backbone is formed with DNA polymerase while it checks for mistakes, and then you end up with two identical DNA molecules, and that is DNA replication. So join me next class with all of your hot questions all about DNA and DNA replication.